I'm Victoria, and I'm working on this project with Joey, and we're doing microarrays. Microarrays were invented in 1991 by Lubert Steyer and Stephen Fodor, founder of Affinitrix, a main producer of microarray components. The uses are to compare levels of gene expression under different conditions, measure host response to viral infection, viral gene expression after infection, and perhaps compare gene expression of two different strains of virus that have infected a host. This is the procedure for doing a microarray. Your first step is to take your cells of interest. You normally get two different samples, and you extract RNA from them. It has to be purified and amplified with PCR. You make cDNA from that with reverse transcriptase. Tag each sample with fluorescent dye. It's usually CY3 and CY5, red and green. Mix two tag samples and pour on the array chip. Complementary sequences will bind together, and then you just rinse off the unbound DNA. Then you load the chip on a microscope slide and put it into an arrayer. The arrayer is a machine that has two lasers, one that picks up each type of dye, and is usually attached to a camera and a computer, so that the images can be saved, and the software on the computer can superimpose each image. See, here is just the image that was picked up by the red laser, the red dye, and then this was the green dye picked up by the green laser. And then the computer superimposed the images so that the places where there are both yellow and, or both, red and green lighting up have turned yellow. So how do we interpret these results? Well, there are four different results. Each individual spot is its own DNA fragment, and it could be black. You might see nothing here. That means that neither of the gene, or neither sample expressed the gene. If the dot is yellow, then both samples are expressing this particular gene. If it's green, only the sample that you tagged with the green dye is expressing that gene. And obviously, if it's red, only the sample that you tagged with the red dye is expressing that particular gene. So what are the time, labor, and cost associated with microarrays? Time is about 1 day if you do the microarray in an in-house laboratory. If you send the samples away from purified RNA to be done further, the time is 1 to 3 weeks for the samples to be analyzed and the report to be returned. Labor could involve maybe just 1 or 2 people. The purification steps go faster with 2 people, but from then on 1 person is fine. And the cost varies greatly whether it is done in-house or at another lab. If you do this yourself, it will cost about $63,000 because the arrayer is $60,000 itself. The software license to superimpose the images is $2,000 a year approximately, and the reagents are about another $1,000 a year along with the gene chip. If you send this out to another laboratory, it will be about $1,000 since they make you pay for the gene chip, the labor, and also you have to still buy the reagents to get through the PCR and amplification steps.
I'm Joey. I'm going to talk about some of the advantages of microarray analysis. One of the advantages is the fact that it's a very high throughput technique. And you can get a lot of information from doing just one test. Also, there's the fact that you can look at a significant portion of the genome at once and examine which genes are being expressed under the conditions that you're interested in. Some of the disadvantages are the fact that it takes a lot of time, especially if you decide to send your results out to another facility. A majority of people actually do that because of the costs associated. And then also, it can be costly even if you do it yourself, because you have to buy all the reagents for all the different tests. Some related techniques for microarray analysis are SAGE, which is serial analysis of gene expression, and RNA-seq,